And I want to thank you so much for the invitation and the opportunity to speak with everybody today. I just want to start by talking about cystic fibrosis, which I'm sure I do not need to for this audience. As we know, it's a disease that starts very early in life, and the fact that your CFTR protein doesn't work probably can affect things even before you're born. And then over time, we see more and more manifestations or complications that are present, including pancreatic dysfunction, sometimes meconium ileus, inflammatory changes, lung disease, and we wind up with failure to thrive and significant remodeling of many tissues, including the lung and CF-related diabetes. And it's been the focus, at least historically, to really, concentrate on the complications that come up in CF and to treat those complications. And we've always had continuous therapy that has really changed and made cystic fibrosis disease that from the early 60s was incredibly devastating and led to an incredibly shortened lifespan, to a disease that, although very complicated, often results in a very robust life. In probably 1989, when the gene was identified, people started to look a little differently at how were we going to really develop a pipeline in therapy. And I think the first 10 years that the gene was identified, there was incredible focus on understanding what the CFTR protein did and how it contributed to disease. And in labs across the country and across the world, people developing potential therapies. But I think that it really was in about the year 2000 that people started to really concentrate on the protein itself and trying to develop small molecules that could make the most of a mutant protein or help repair CFTR. And I think in the last 10 years, when we look at the therapeutic pipeline, you can see that there's been incredible focus there. And it's really managed to come up with a number of new medications that have been, in some cases, life-changing for people. Let's spend a little bit of time to look at that. And that's been really the focus in the last 10 years. And it's, again, life-changing. But I think that there are definitely patients who have CF that are not going to find that some of these therapies are terribly effective for them. And then when we think about what would be our next big step, what's our next approach that's going to be game-changing, just as the protein correctors, potentiators, amplifiers have been game-changing, where do we go from here? And so as we look to where our next big push is going to be, I think we need to look at other types of therapies that we haven't really looked at before. And those include RNA therapies, which are therapies that can be given periodically and we can restore CFTR protein. And then ultimately looking at gene therapies where we can have permanent repair, restoration and repair of the CFTR gene. And we're going to spend a little time to think about where we are now, how we got there, and where are we going and what's emerging. So if we go back to where we've been, so repairing CFTR, we really need to think about how did this all come about? How did we all of a sudden get all of these wonderful new drugs? And I think the way we really were able to take that leap forward were when scientists who were in academic labs really put their heads together as a community and said, we probably need a different platform. And what was developed was high throughput screening, where 10,000 compounds could quickly be tested in medicinal libraries. And robotics came into place so you could make a detection assay to really look for if you could see if CFTR could function. And this needed to be reproducible, and these efforts began around the turn of the century, the year 2000. And these are just robots, and these are assays that can really identify compounds. And you know, these were sort of game-changing, because up until now, people would identify one compound, it would look very promising, we test it, and then, oh my goodness, there was often disappointment. But now you could just test libraries after libraries after libraries. And I think, you know, there was a huge academic push in the labs of people like Dr. Verkman, and then from a pharmaceutical and biotech standpoint, Aurora Pharmaceuticals really started to develop these things in around the year 2000. And Aurora was eventually bought out by Vertex, and that has led to drugs that we now use in the CF community that have been really quite impressive in how they change people's daily lives. So there's been Kalydeco, which came on board in 2012, Orkambi, Symdeko, and then last year there is Trikafta, which has been a remarkable drug for a number of patients. I just want to take a minute to highlight something out of the New England Journal that was from the original trials for Trikafta, which is the brand name of elexacaftor, tezacaftor, and ivacaftor. So it's a drug that has 3 components. And I just want to point out here, changes in FEV1. Here is somebody who's on placebo, or these are the patients on placebo. And here's what happens when you get the drug. It's a 13% improvement in FEV1. Sweat chlorides dropped by 45 millimolar. Numbers of exacerbations also decreased significantly, and quality of life seemed to improve dramatically. So this is a drug that now can be given to patients who are homozygote for the most common mutation, F508del, or who have a single copy, for anyone who's above age 12. And so it's really been pretty life-altering for many patients who are able to take this drug. And so I think that this shows the power of things like high throughput screening if you can develop an assay and you know what your target is and what you want to do. And it's all very, very exciting. You might say, Marie, we can stop there. We have a drug that really is going to work for people. But we also know that there's a lot of heterogeneity in responses to drugs. So not everybody responds the same way. And so in the pipeline right now, there are still a lot of small molecules that are being developed. And these are just 5 of the small molecule programs that are in clinical trial in phase 2. And so again, I think that it's incredibly important as we see the success with some of these drugs to not put all our eggs in one basket, and to make sure that we continue to develop drugs for patients, because not everyone's going to respond the same way. And we still have many other mutations that are not served by these small molecules. So all of that small molecule work is really great, but it only works for people who have a CFTR protein that can be repaired or can be optimized. And so if you have other mutations like nonsense mutations, stop mutations, then those small molecules are not going to work for you. So how do we get people who have nonsense mutations, small molecules, and what's the goal for those? So for those, we need to develop a new assay that allows us to look for read-through to restore CFTR. And there are a number of programs that are in the pipeline. Almost all of these are preclinical, except for Elox Pharmaceuticals. And they're looking to develop and identify new compounds that would allow for read-through of a stop mutation, which would restore CFTR protein. There will be challenges here, just as there are challenges with small molecules. You got to look for specificity and toxicity. But again, there are things that are in the pipeline in the preclinical space, and then Elox Pharmaceuticals has a phase 2 that's ongoing. So those are all very exciting things. The things to keep in mind are that for small molecules, for some of the other mutations, we've been working on that for 10 to 20 years. And so for nonsense mutations, we haven't really applied this high throughput screening approach in such a robust form in the past. So we probably have some catching up to do. Right, so that's about small molecules, but what else is in the pipeline that will really be earth-shattering and paradigm changing and shifting, just like high throughput screening was paradigm shifting 20 years ago? Well, first we're going to talk a little bit about RNA therapies. So what are RNA therapies? Well, RNA is nucleic acid, sort of like DNA. It comes in a bunch of different flavors, though. There are a bunch of different types of RNA. There's messenger RNA, which is known as mRNA. There are small pieces of RNA you can design called antisense oligonucleotides. And there are tRNAs or transfer RNAs. There are an array of other RNAs as well, but these are the three main types of RNA that are really being worked on in the CF space to look for potential therapeutic targets. So mRNA, or messenger RNA, is a very interesting approach to potential new therapies, because if you were to use mRNA, it could benefit everybody who has CF. It's not mutation specific at all. And so when we think about RNA therapies, I think that one's pretty cool. Antisense oligonucleotides, they can be designed to help in a variety of manners, and we'll look at them as an example with splicing mutations. And then transfer RNAs, well, those are RNA therapies that might be really helpful for people who have nonsense mutations. So let's take a look. First, messenger RNA replacement. All right, so don't be alarmed. In the graphic on the right, we have it's a cell, so we're going to be back in Bio 101. And if we go to the bottom of the slide, we can see this coiled DNA, or the blueprint of who you are. What happens with that blueprint is we make a messenger RNA, which is basically the instruction booklet. The instruction booklet then makes a protein, and the protein goes through this part of the cell called the endoplasmic reticulum. This protein, CFTR, then gets packaged and it goes to the surface of the cell where it works. So what happens if we deliver a messenger RNA to the cell, bypass this whole gene and your own mRNA, and just give enough messenger RNA to the cell so that the cell can then start to make CFTR protein? Wouldn't matter what mutation you had, whether it was a stop or anything else, we could deliver the proper CFTR message and we'd make the proper protein. So that would be very cool. The biggest issues with trying to get messenger RNA into a cell is delivery. So in this graphic, you see that this thing that looks like a spaceship around our little messenger RNA, and that's a delivery system. And so we'd have to come up with appropriate delivery systems to get messenger RNA into the cell, so then you can make a protein and get CFTR to the surface. So you might say, okay, that's like, that's science fiction, Marie. But actually, there's already clinical trials that are ongoing. One is called Restore CF. You can see it on clinicaltrials.gov, where there is a specialized carrier to take messenger RNA into the cell. so that you could make CFTR and get it to the surface. Potential limitations, we have to figure out how long this would last. Could we really get a good result where maybe you could inhale this and we could see changes in lung function? If you wanted to fix everything, how are we going to deliver that? Is the same delivery system going to work if we deliver it to the lung as if we try to deliver it to all the cells in the body? And you have to worry about or think about inflammation over the long term. What about antisense RNA oligonucleotides, or ASO therapies? So, okay, I told you before, these are little parts of RNA. They're single strands. They can be designed to block a messenger RNA if you wanted to degrade a messenger RNA, to upregulate or enhance a messenger RNA. There's lots of things these little ASOs can be made to do. In fact, this is a technology that's already in the clinic. There's already FDA-approved therapies for different diseases: retinitis, muscular dystrophy, spinal muscular atrophy. So how would these be helpful in CF? Well, one way they might be helpful is if you could design a little ASO that might help block or camouflage a splice site that is created when you have certain mutations in CFTR. So we know that there are certain CFTR mutations that, when the mutation is present, suddenly a splice site is developed in the messenger, in the gene, which leads to a messenger RNA that's not normal, and it leads to a non-functional CFTR. If you design one of these little ASOs to block that cryptic or unmasked splice site, suddenly, instead of the cell identifying something that says, cut me here, and then making a non-functional protein, now with that little ASO in place, you can't, the scissors or the molecular scissors of your cell can't see that, and you make a normal messenger RNA and normal CFTR. And this is all work that's still in the preclinical realm, but the ASOs that have been designed to work on certain mutations, when they treat patients' epithelial cells from the nose, so respiratory cells, they can lead to normal messenger RNA and normal CFTR and normal function. So this is a very exciting approach, and we all hope to see this develop into something that's going to be clinically possible for our patients. And again, I think we should have hope because there clearly is, there are other ASOs that have been developed for different therapies. Okay, the third RNA therapy that we might want to consider in CF, well, that would be transfer RNA therapies. What are transfer RNAs? Again, we're going to go back to Bio 101, and I apologize, but I think these are important concepts. We have a messenger RNA, and if you have a nonsense mutation or a stop mutation, your messenger RNA or your instruction booklet all of a sudden says stop right in the middle of the instruction booklet to make the protein CFTR. This is the ribosome. The ribosome is the protein making factory. So normally you have this little instruction booklet, and what happens? In the instruction booklet, transfer RNAs are specialized RNAs that bring the amino acids, which are the building blocks to the protein, to the protein building factory, and we make this long protein chain. This is the beginning of CFTR. As we are marching down the instruction booklet, putting in these little transfer RNAs and bringing in the little amino acids, all of a sudden we hit stop. And what happens? We stop making a protein. And we have a part of a protein that's made that's not functional, and then we don't have CFTR. So what happens if you could engineer and design a transfer RNA that could read this stop, but instead of stopping, it adds an amino acid? So you would engineer this tRNA to be a little different. It would deliver an amino acid instead of stopping the protein production. And then you'd continue to read right through that, and you'd make a full-length CFTR. So this has been done in the lab. You can see on the right-hand side, okay, this gray area is a gel. These black lines here by the arrows are full-length CFTR. Here's wild-type full-length CFTR. And what you can see is this is somebody with a G542X mutation, and then here with a W1282X mutation. And you can see there's full-length CFTR that is made when the addition of these engineered transfer RNAs are added to the cells. So again, incredibly exciting work, and this is done out of an academic lab. And there's a company called Recode Therapeutics that is working on this, and it's in the preclinical stages. So again, this is another potential therapy that would be very exciting if it continues to look this promising as we move forward. So those are the 3 RNA therapies that we really think about, that are in our pipeline and that are being worked on, and hopefully will wind up being therapies, not tomorrow, but down the line. But now we're going to change our focus a little and look at gene transfer therapies and gene editing therapies. Okay. So gene transfer therapy. I say, gene transfer therapy, what's that? Well, it's also otherwise known as gene therapy. It's when we add a normal copy of CFTR to the cells of people who have CF. So in the end, you wind up with 3 copies of the gene, 1 healthy, and the 2 that you have that have CFTR mutations. So gene therapy is not a new concept to this community. We've done gene therapy in the late 80s, early 90s. I shouldn't say late 80s. But I remember being at the the CF conference in 1989, and there was a very young researcher who was introduced at one of the plenary sessions to tell us about gene therapy at adenoviral vectors to deliver CFTR to cells once the gene was identified. There was a decade or more of incredibly beautiful work and many clinical trials trying to make sure that we could deliver CFTR and that we could cure CF. And I think what we did is learn an incredible amount during that 10 to 15 years, but the tools that we had at the time were just not sophisticated enough or robust enough to lead to a clinical outcome that led to cure. And so what's different now? Well, there's a lot more work that's been done. Some of the tools are much more sophisticated. And in the late 80s, early 90s, we were probably the pioneers of a lot of gene therapy. And now there are a number of other folks and other scientific communities that have been working on this as well. And it's actually led to FDA-approved gene therapies. So the concept that this can happen is there. So there's gene therapies for cancer, inherited retinal diseases, beta thalassemia, and spinal muscular atrophy type 1. And we're going to spend a little minute to talk about SMA type 1. And I say that because it's a devastating disease. I'm a pediatric pulmonologist by training, so I take care of children all the time with lung disease, and so SMA is something that is a disease that I also see at times, And I think it's a very devastating illness. It's a disease in which children, when they're born, they're healthy, and then over the first few months of life, they start losing milestones. And it's because the protein product, SMN, is essential for motor neuron health. So that's all, it's the it's the nerves that help move your muscles. And so that gets destroyed. But there is in the last year, a gene replacement therapy that was approved by the FDA. It's a one-time injection. And when patients or subjects were given this gene replacement very early in life, the first few months of life, suddenly a disease that often led to death by age 2 or 4, or ventilator dependence in the first year of life, has changed dramatically, so that patients no longer need to be mechanically ventilated, and that they, depending on the patient, can be running around and dancing at age 3 and 4. So a game changer. So clearly, gene transfer may indeed be possible. So how is it that the folks with SMA were so much more successful, and what are the challenges in CF? So the SMA gene is smaller than the CFTR gene. So it was much more easily encapsulated into an adeno-associated viral vector type 9. It could be delivered to these neurons that are long-lived and don't replicate, which is different than in CF. If we think about just the lung disease, we have to deliver a very big gene to a complicated tissue. And these are cells that, turn over, they replace themselves. So we're not talking about just delivering it to the top cells here. We got to deliver it way down here to the basal cells. And when we think about CF, we're delivering it not only to the tissue in the lung, but to many other tissues. So for gene therapy in CF, some of the biggest issues are delivery, right? Delivery. And I think we've all been living through delivery in our own lives on a daily basis. So what do our delivery systems need to look like? And what are the delivery systems for gene therapy? Well, I think right now, you should know, in a preclinical world, they're looking at AAV vectors, lenti vectors, non-viral vectors. So there's a whole array of different delivery systems. And they're designing these delivery systems, so they're not just counting on, in the past, they might count on a virus, capsid, or a capsule on a virus, and put our gene in that virus and then deliver it to cells. But now they're actually designing their very own viral capsids and looking at the best of all different viruses to see what's going to be most effective for different tissues. So it's a very exciting time. There are gene therapy trials that are being designed and queued up in the UK for the near future. And I know that that's also going to be something that we'll see in the US in the future as well. Now, there's also gene editing. So what's the difference between gene transfer and gene editing? Well, gene transfer or gene therapy, we give you an extra copy of the gene. For gene editing, we repair the mutation that's in your own DNA. So there's a lot of ways to do this. There's zinc fingers, there's CRISPR, there's TALENs, there's PNA DNA. There's a whole bunch of different platforms. CRISPR is the most common. And you should know that when it comes to CFTR, all of them have been tried on CFTR and tried to correct mutations, and all of them have worked in an array of different cells and animal models and cell lines. But the take home is that CFTR can be edited with all of these different technologies. So that is very exciting. So how would, you might say, okay, Marie, you showed us that in gene therapy, there's already FDA approved stuff. What about with gene editing? With gene editing, we don't have approved therapies yet. We do have clinical trials though. And one of the most promising clinical trials is in sickle cell disease. And I'm going to use that as an example here. So for sickle cell disease, we got a single mutation in a beta globin gene. The mutated protein causes your red cells to sickle. We got a whole bunch of damage that then happens when the cells sickle. And the only therapy right now for people with sickle cell disease, the only cure, I should say, is a bone marrow transplant, and that isn't a possibility for a lot of people. So there's a gene editing clinical trial in which patients started to get CRISPR-based gene editing for sickle cell disease. And I can say that the interim reports are that patients who've received this are doing quite well. So again, when we think about correcting sickle cell versus correcting CFTR, we would say, okay, Marie, if they can do this for sickle cell disease, when are we getting this for CF? Well, remember, for sickle cell disease, everybody who has sickle cell disease has the same mutation. So when they had to develop reagents, it was one set of reagents, one technology. For CF, we have a lot of mutations. And for each mutation, you'd have to have each mutation requires its own gRNA, its own little designer set. So as you can see, this is going to be a much bigger fix for CF than it is for sickle cell disease. The other things to keep in mind is you still have to deliver all of the reagents to the cells. You have to know which cells you need to deliver it to. And my goodness, we're dealing with more than one tissue. So can it happen? Sure. We know it can happen because we're seeing it happen for other diseases. But again, our challenges are going to be a bit more. So what else do we have in our pipeline? There's cell-based therapies. And I'm actually not going to talk to you about this today because tomorrow at 10 o'clock, you have a wonderful session on gene-corrected autologous stem cells to treat cystic fibrosis by one of your investigators from Stanford University, whose work is just awe-inspiring. So I'll just leave that tantalizing message that you really should cue in for tomorrow at 10. So just to recap, we have many novel approaches to get to the heart of the problem, many advances in the field that'll make these therapies feasible. And there are some of these novel therapies like mRNA therapy and even antisense oligos for other diseases that are already being transitioned to the clinic. So it's an exciting time. Challenges, as I say, challenges are multi-system disease. We have the respiratory tract, the GI tract, pancreas, sweat gland, vas deferens, immune cells. So delivery may be different depending on the tissue. And again, in this day of COVID and home delivery, it may be a DoorDash versus Grubhub versus Uber Eats vector that delivers things to each different cell type. We need to understand how are we gonna approach this? Do we just do the lung disease? Do we really think about cure as all tissues? How do we target those? What's going to be different? And how do we keep this safe with low toxicity and off-target effects? And in the lung, there's also, well, in the GI tract, there's the mucus. And how do we get around that? Do we deliver from above? Do we deliver from below? As far as the cells are concerned, from the blood side or from the lumen side? Lots of things for us to be able, that we really need to figure out. And targeting airway progenitor cells. Basal cells, this is a slide you saw before, are these cells on the bottom in the red box. So targeting those cells is a lot more difficult than targeting the surface cells. We're going to have to target these from the blood side, because I think it's going to be much harder to disrupt this layer and get to the basal side. And so it's gonna take a bunch of work and a lot of creativity, but we're a community that is filled with both. And in the end, the goal is for everybody to have a therapy to prevent disease. It's going to take a community, the CFT members, your families, industry, scientists, clinicians, and we're all going to have to partner together and work through this. I used to have a slide between this slide and the last with a timeline about when could you expect any of these nucleic acid or genetic therapies to be coming to a theater near you, to your TDN site. When were we going to see this? And I have to say that I took that slide out because right now I don't know, and I don't know because in the times that we're in right now, a lot of the research that we've wanted to have move forward has stopped for a bit. I think all of the research I know where I'm in Connecticut, is started back up, and so we're totally jazzed, and I think that's great. But I do think that we're going to wind up with a little delay in clinical trials just because we need to keep people safe, and we can't have people starting a clinical trial of a very experimental drug if we can't have you come to a clinic site to be monitored closely. So we'll have to see. In the past, it might have said 3 to 5 years. We might have to push that out just a little. I want to thank you so much for listening. Have a wonderful day.